In the last video, this is where I left you, and I said, hey, guess what? In a manufacturing process world, there are two routes. One route needs Mother Nature, and the other route does not. I can kind of do my stuff and my synthesis stuff and make things, and I don't need the help from anybody else. I can do it. Just give me the raw ingredients, and we'll put them into a vat. We'll mix, we'll stir, we'll heat, and then we'll isolate them. No big deal. I slap my hands, and woohoo, I'm done. All right, well, in the biomanufacturing world, it's a little different because we're relying on things to do a job for us. And those things can be a number of different options, but they're biological in nature. Hence, bioprocessing, biomanufacturing. That's what this is all about. Hello. All right, so the very first thing that we need to talk about is the fermenter and bioreactor that we see in the very beginning of this flow chart. So right here, biotech product. Yes, we got one. Okay, so fermenters and bioreactors are very common. I'm going to probably see those at some point in time. What is it? What are they doing? What's happening on the inside? Well, that's kind of the purpose of this video. So the first thing that I want to introduce you to is to a term, and I told you two terms are coming up. One is upstream and one is downstream. So I need to introduce you into, ta-da, upstream. All right, so upstream basically means biological material selected and grown for manufacturing. That's what upstream is. And I will say that out of the entire manufacturing flow chart that you're seeing here, upstream is the smallest portion. It represents the smallest piece of the flow chart, but it's very important because if I don't do this the right way, everything else fails. So upstream is very important, even though the majority of all the other steps that are seen here on this flow chart are what we would call downstream processes, not upstream processes. So upstream looks like it's the biological material selected. So we got to pick out the right stuff to do the right job, and grown. So I got to grow them. I got to nurture them. I've got to be the parents of all of these little things that are going to grow and make little colonies and populate. All right, so I'm going to grow them. And once they're grown, then they need to be used. So this is the part of upstream processes that we need to talk about. So the very first thing down here in the very bottom, fermenter, bioreactor. What are those things? Well, here's a schematic. And I'm just going to kind of read this little blurb and we'll talk about it. It says an apparatus that maintains optimal conditions for the growth of microorganisms used in large-scale fermentation and in the commercial production of antibiotics and hormones and other biologically derived pharmaceutical compounds. All right, so in other words, what this means is that this fermenter, this bioreactor step, folks, this is my aquarium tank, okay? I've got an aquarium tank. There's fish on the inside. It is my job to take care of that aquarium tank because I don't want my fish to die, right? I want my fish to prosper. I want my fish to grow a long, healthy life. I want my fish to be happy. So I'm going to constantly go to my fish tank and I'm going to check all of the conditions. I want to make sure that it's clean. I want to make sure that the pH is kind of at the appropriate level. I'm probably going to test my chlorine. I'm probably going to test my dissolved oxygen content to make sure my fish has everything that they need. I'm going to feed them so they can continue to grow, and then they might have little fish babies, and then if they have little fish babies, I get really happy, and then the colony multiplies, right? That is what's going on in the fermenter and bioreactor step. This is the fish tank of the entire process. That's what's going on. All right, so that means on the inside of this thing, there I see it. It's in the bioreactor. It's in the fermenter. There's stuff on the inside, and that stuff is growing, and we need to nurture it. We need to be the mummies, and we need to be the daddies to all of those little algae that might be in there, or those colonies, whatever they might be. I need to make sure they have everything that they need. So I am the caretaker. So I'm going to constantly look at them, and I'm going to constantly monitor them, and I'm going to constantly make sure they have everything in place, so that way I'm not a bad one, and that way I don't kill everything because nobody's going to ask me to take care of those fish again. And guess what? If I kill colonies out in the biomanufacturing plant because I don't know what I'm doing, no one's going to ask me to come back for the job again because I have just destroyed probably their little gold nugget. Because without this, guess what? You can't do anything because without this help, without these things actually doing what they need to do, I'm not going to get a product. And if I don't get a product, I can't sell a product if I'm a company. So with that said, a couple of things. Look, it's not surprising. It looks like I've got some intakes. Look, nutrients. So I got a little place that I can add some nutrients, and then all of the things on the inside here go yum, 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 yum. And they just continue to make babies, and they continue to grow, and they continue to get big. And then we see them off to college, and we wave our hands and go, bye, don't come back for a while, see ya. Okay, well, there's some nutrients that are going into the vat. Something else is acid and base. So there are chances that I need to control the pH. So if it goes too acidic or if it goes too basic, I need to quickly adjust that back down into the optimal pH level that we need to do. So I'm going to have an intake line, and that intake line is going to control the acid or the base levels. In other words, the pH that these processes will need in order for them to flourish and for them to grow. That is what we're after there. So thank you, acid and base, and thank you, buffers. Next up, okay, as these things grow and as these things do what they need to do, there is a chance that they are generating waste. And we can monitor that waste. We can take that waste out. But I've got to have a way that I can kind of handle this and do this without disturbing everything or killing everything in the meantime. Notice there's a little pH probe that goes in here that tells me what the pH is. So that way I can go up here and adjust for the acid and base intake, depending on what we need. There's also a temperature probe here as well. So it constantly gives me the temperatures inside of the tank. So that way, if it gets too cold or if it gets too hot, I can kind of monitor that as well. Imagine a fish tank again. If that fish tank goes too cold or too hot, well, you're going to have some cooked fish at the end of this, or at least some dead fish if you're not very careful. We get a sparger. A sparger just just bubbles some air in. So maybe that sparger is going to kind of give us some dissolved oxygen that needs to go into that vat, or some type of gas that we need to introduce into the vat in some form or fashion. Over here to the left, I see some cold water, a cooling jacket, an oxygen probe, an impeller. What is an impeller? We'll talk about those types of things. Sterile nutrient medium. Ooh, that sounds fancy. Again, we'll talk a little bit about that as well. Some steam that can go into the bioreactor or the fermenter. And then we see some things up here at the very top, some antifoam and a rupture disc and a tank vent and some pressure gauges. Well, okay, all of these things we're going to have to dissect. We're going to have to talk about them in little bitty pieces, but I hope that I give you an idea of what these fermenters and bioreactors are basically doing. These are big vats, and these vats have living things on the inside of them. And we have to take care of these living things to make sure they stay alive, so that way they can keep reproducing, they can keep multiplying, and they can keep producing product that we are after. All right, so with that said, there's a little schematic. What's a schematic? All right, well, give me a real picture, Tracy. I'll show you a real picture. Here is a bioreactor, and this says pilot scale. All right, so what these things are, they can be very tabletop friendly. So bioreactors and fermenters can go onto a tabletop, and those tabletop can be like testing grounds. Let's see if we can do what we need to do with it. And we're not going to destroy, hundreds of liters of stuff. And instead, we're doing very tiny scales where we're working with maybe a liter instead, and trying to figure out the best optimal conditions to grow these things. All right, that's research and development. So those can be bioreactors, those can be fermenters, but they can easily go above and beyond the tabletop. And here you see see them sitting on the floor. And here you see a big old vat, right? There we go. And that big old vat has some stuff in it. And that stuff is alive, alive, alive. It's like Frankenstein, right? So we want to make sure that those stay alive, and we have to monitor different conditions in order for them to do that. What you see on the screen right now are 4 of the most important things that we need to look for, that we need to monitor, that we need to double check. The first one is temperature. Again, we want to make sure Goldilocks, that's what we're after. Not too hot, not too cold, just right. And let baby bear come back home and figure out what's going on. All right, number 2 is pH. Again, we need Goldilocks, so not too acidic, not too basic, but just right for that particular colony that we are trying to grow. Number 3 is dissolved oxygen. So dissolved oxygen, also abbreviated as DO. DO is basically oxygen gas, and you need oxygen gas to breathe, and you need oxygen gas in order to function. And you know, there's lakes and there's rivers and there's streams that have dissolved oxygen in them. Fish need it in order to kind of do what they need to do and grow and be happy. Well, okay, this can get out of whack. So we have to make sure that we have enough dissolved oxygen in the system for that particular colony or that particular run that we are worried about. And then finally, number 4 are nutrient levels. So nutrient levels is the food. Yum, 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 yum. So we got to make sure that these things have enough food in order to stay alive. And that means I have to add over a course of time to make sure that that supply does not run out. Okay? So for instance, I'm going to give you an example here. Let's say that we are trying to make our own moonshine. Moonshine, woohoo! We're going to make popcorn Sutton happy today. All right, so in that particular flask or container or barrel in the middle of the mountains, I'm going to probably put in some yeast. Oh my gosh, guess what? That yeast is a biological molecule. That's what that thing is, right? It's alive. Okay, so that yeast is going to do a job. And I need to make sure that yeast stays active and it stays alive and it has the nutrients that it needs. That means that the process of making moonshine is very much like a bioreactor, and it's a bioprocessing technique. That's what this is. So I get yeast, and what I normally do is probably add some sugar. Yum, 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 yum. All right, well, that yeast is going to eat the sugar, and it begins to ferment. And the yeast begins to do something. And that yeast begins to poop and pee out CO2, which is carbon dioxide gas. Oh, okay. Well, these are inputs. Ding, 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 ding. That's a term I already know. And then it's going to give me outputs. And part of that output is going to be CO2 gas. Bubble, 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 bubble. And as long as that tank is bubbling, then that means CO2 is getting made, and that means that the yeast is doing the work. All right, well, imagine this stainless steel big container right here, completely sealed off. No leaks. And that yeast is going to generate CO2 gas. What's going to happen to the pressure? What's going to happen? It's going to go up, 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 and then before you know it, there she blows. That's what's going to happen. So I need to go through and I need to make sure I'm constantly monitoring the pressure, because CO2 or other types of gases can be basically given off, and I need to vent those out in some form or fashion to prevent an explosion from happening inside of the environment. Next up is the sugar. These are the nutrients, right? These are nutrient levels. Hello. Those nutrient levels, that's the food, it's going to get used up. So I need to make sure there's enough of that. So I need to be able to go in and add more sugar if I need to add more sugar in order to make more product in the very end of this. I need control of that. I need to be able to take care of this yeast so that way I can get really good moonshine and sit back in the mountains and get drunk. I mean, that's what this is all about, right? All right, well, another part of this is that it farts out CO2, but guess what? It's going to pee out a little bit of alcohol, ETOH, that's ethyl alcohol. That is the abbreviation that we like to use in a lab environment, ethyl alcohol. And that ethyl alcohol, that pee out of the yeast, is what I'm trying to harvest and what I want to drink. Oh, think about that the next time you take a shot of a whiskey or some type of vodka or tequila or whatever you decide to drink. Maybe you don't want to drink at all, and if so, that's great too. But the alcohol is the product of interest. That's our target. That's why we're doing all of this in the beginning. So now I get this big vat, and in that vat I've got yeast, and I've got sugar, and I've got CO2, and I've got alcohol, and I got to figure out, after this yeast does what it needs to do and produces this alcohol that I'm after, how can we get that alcohol out of that tank? How can we clean it up? How can we transfer it over? How can we just make sure it's alcohol only that I'm getting in the end, because I don't want all the other stuff that comes along with it. These processes are all different depending on what we need to try to make. But this is a bioreactor. I mean, that's kind of what's going on. I mean, think of it that way at least, right? We are allowing a living organism, yeast, to make a product. So inputs and outputs and the processes that go along in the meantime in order to get to that final product that we are after. So a couple of things that I just want to clarify before we go to the next video. First off, we do have reactors, and then we've got bioreactors. There's a slight difference between the two. So if I look at this term reactor, what that means is that those are just used to grow cultures. That's what's going on there. So we don't necessarily use that for any other purpose. And the reason is because we just don't want to kill out the entire colonies, right? We might want some backup here. So I've got these big reactors, and these reactors are just dedicated to growing. That's all that they are doing. So nothing synthesis is going on in them. I'm not make a product at all in these reactors. These are just big old vats that we are keeping stuff alive in, and that is it. So once we get so much, what happens is that it is transferred into a different container, and that different container is another reactor, but that reactor is going to be doing stuff. And because of that, that reactor is called a bioreactor. And that's what this down here is basically saying. Listen, a bioreactor is going to be a vat, and that is going to create this kind of synthesis that you need in order to make a product. And the bioreactor is what you are adding stuff to. You know your product is in there. It's going to have to be separated. It's going to have to be purified. It's going to have to be manufactured in some point or form or fashion. But the reactors are kind of like the original holding tanks. That's what those are. So reactors are growing the stuff. And once we get so much, we're going to transfer the material out of a reactor. Not all of it, but some of it, right? Some of it's going to be transferred into a different reactor. But that reactor is a little special because that's where magic is happening. That's where things are getting made. So that bioreactor is going to have to be nestled and taken care of a little bit more, because sooner or later, that's where my active pharmaceutical ingredient is going to be. And that could be a number of different things. But that is basically the magic pot that we need to isolate and we need to take care of from that moment on. Bioreactors, those are mammalian cells. Those are cells like with nuclei or nucleus on the inside. I'm not going to go down that rabbit hole in biology too much. But if it's not bacteria or if it's not yeast, it's probably mammalian in nature. We do have two different terms that we have dedicated for this. So a bioreactor would be basically the mammalian cell that is, for the most part, what we see very often. The fermenter, though, this would be bacteria-based or yeast-based. So I want you to think of a brewery, right? A brewery is probably using some type of yeast, and that yeast is doing a job, right? So when I talked about making moonshine, or when you go down to Front Street Brewery, you are probably looking at these big fermenter tanks. Those are not called bioreactor tanks because it is a different type of source material on the inside. If it's yeast or if it's bacteria, those are fermenter tanks. So that stuff is making a product, and that product we're going to eventually isolate and purify and clean up. Okay, that is kind of the same notion as a bioreactor, but the difference is that in a bioreactor, these are actually mammalian cells that are made and structured in a different way. So that one of the core differences is that those cells have a nucleus on the center, and that nucleus inside of the cell, that nucleus is going to house DNA and those types of things, the majority of the cell contents at least, on the inside. So those are the slight differences of the terminology. Very often, overall reaching goal, they are kind of the same, but they are slightly different based on what they hold on the inside. All right. So this is what we call the upstream portion of bioprocessing work. It's biological material that you are selecting, that you are using, and that you are growing eventually for the manufacturing line. So what this means is that the fermenter bioreactor step right here in the very front, this for sure is going to be part of the upstream process. Now, in the upcoming video, we need to talk about, okay, how far does upstream go? Like at some point in time, we get out of the upstream and we go to the downstream. So what step does that happen in? That's what we need to discuss. And that is going to define downstream for us. And we'll learn about that in the next video. So again, come back and see me. Keep learning. I hope that you do. That's why you're taking the class. And as always, if you got questions, you can let me know. But keep taking notes, keep studying. I know that you know more now than what you did when you pushed the play button. So keep pushing the play button and keep learning. That's what this is all about.